Hello, good morning. Today I want to discuss deviation of Mendel's monohybrid ratio, also about incomplete dominance, codominance, multiple alleles or factors, and lethal genes, which deviate the Mendelian monohybrid ratio. Before discussing deviation of Mendel's monohybrid ratio, we should know what is monohybrid cross and what were the results of such cross and ratio. Monohybrid cross is a cross when only one trait and its two alternative forms are considered during hybridization experiment. For example, if we take red flowered male plant and is crossed with white flowered female plant or vice versa, it will be known as a monohybrid cross for color of flower. Now this diagram shows that purple flower, white flower crossed, F1 will get in F1 we will get all purple flower plants, and in F2 purple and white will be segregated in 3:1 ratio, that is the phenotypic ratio. Now this was Mendel's observation. Now it can be genetically expressed like this. The parents are capital R, red, one of the parent, other is small r, that is white, and the gametes will be capital R, small r. The hybrid will be capital R, small r, and all the plants will be red due to complete dominance. F1, the gametes will be allowed to self-fertilize. So in F2, the combinations will be capital R, capital R, capital R, capital R, small r, and small r, and the ratio will be 3 is to 1, 3 red and 1 white, and the genotypic ratio will be 1 is to 2 is to 1, that is capital R is to capital R is to small r. On the basis of results of F1 generation of monohybrid crosses for all the 7 traits, Mendel proposed that in a cross pure for contrasting or alternative forms of a trait, the one which is expressed itself in F1 generation is dominant and is able to suppress the expression of other or recessive form. In F1 generation, all plants bear red flower. On the basis of results of F2 generation obtained in monohybrid cross, Mendel proposed first law, that is law of segregation, which states that when a pair of alleles are brought together in a hybrid, they do not mix up or blend but remain associated with each other and separate or segregate from each other in next generation when hybrid forms gamete. So in F2 generation, both red and white flowered plants appeared in 3:1 ratio. There was no blending or mixing up of traits. Now we will see what is the deviation of monohybrid ratio. This cannot be called as interaction. So on the basis of results of monohybrid cross, all the plants with red flowers in F1 generation, Mendel concluded that of two alleles for a trait, one which is dominant appears in F1 and in its presence the recessive allele cannot express itself. So in F1 both capital R, and capital R small r appeared red. But in some cases, results are not like this. In exceptional cases, all F1 plants are with pink flower, that is, intermediate between two parental colors, red and white, and F2 phenotypic ratio obtained was 1 is to 2 is to 1 for monohybrid cross instead of 3 is to 1. Example for this incomplete dominance is can be seen in Mirabilis jalapa and Snapdragon mazes flower color. This is known as incomplete dominance. In these plants, when a cross is made between a red flowered plant and a white flowered plant, F1 generation showed pink flowers instead of dominant type red. Dominance in this case is incomplete, and F2 generation showed one plant with red flower, two with pink flowers, and one with white flower. So phenotypic ratio becomes 1 is to 2 is to 1 instead of 3 is to 1. The dominant allele that produces the red color does not suppress expression of other allele, which is responsible for white color production. So pink color is resulted. Now in this slide we can see that their parental plants, one is having red flower and other is having white flower, and in F1 generation all the flowers were of pink color instead of red as that of Mendelian inheritance. Now it can be genetically represented in a bit different way. Instead of taking parents red capital R, for white we have used WW, and parental gametes are capital RW. Hybrid is capital RW. This the F1 generation plants are with pink flowers, not red, means both red as well as white color is being added. And in F2 generation, the results are red, pink and white in ratio 1 is to 2 is to 1 instead of 3 is to 1 of Mendelian inheritance. Now this diagram again shows the diagrammatic representation, and we can see that in F1 all plants are pink. In F2 there is red, pink and white in the ratio 1 is to 2 is to 1. An offspring's phenotype is intermediate between the phenotype of its parents. Now let us see what is the difference between dominance, incomplete dominance and codominance. Dominance is which completely dominates. Phenotype of heterozygote is same as one of the parental homozygote, no blending. As in case of Mendelian traits, incomplete dominance, phenotype of heterozygote is intermediate between phenotypes of two parental homozygotes, that is red and white. It mixes up to give a pink form. Codominance phenotype of heterozygote includes the phenotype of both the homozygotes, means both are contributing equally. Both are codominant. Incomplete dominance is a form of intermediate inheritance in which an allele for a specific trait is not completely dominant over the other allele. This results in third phenotype in which the expressed physical trait is a combination of dominant and recessive phenotype. In codominance, an additional phenotype is produced. However, both alleles are expressed completely. Incomplete dominance is similar to but different from codominance. In contrast, Mendelian traits show complete dominance. Capital R allele suppresses the expression of small r, so capital R small r heterozygote appears red. In this case, only dominant allele contributes and recessive allele remains silent although present. Now let us discuss about multiple alleles or factors and codominance. Now, up till now, we have studied that one trait is controlled by one gene, and this gene has two alternative forms or alleles. Each is responsible for a allelomorph. But sometimes there are more than two alleles. These are known as multiple alleles. Many genes have more than two or multiple alleles, even though one diploid individual can only have at most two alleles for any gene at a time, such, such as ABO blood group in human, which is an example of multiple alleles. Multiple alleles may result from the different mutations of the same gene. In this case, it is the gene I responsible for blood trait. Human ABO blood types are determined by alleles A, B, and O. A and B are both codominants over O. The only possible genotype for blood type O is OO. Both alleles are OO. Type A blood group can have either AA or AO genotypes. Type B blood group can have either BB or BO genotypes. Type AB blood group has only AB heterozygous genotype. The A and B alleles of gene I produce slightly different antigen A and B on the surface of blood cells. Homozygous A individual have only the antigen A. Homozygous B individual have only the B antigen. Homozygous O individual produce neither antigen. Type AB individuals produce both A and B antigens. A and AB type has both A and B antigens and are therefore universal acceptors. O type has no antigen, so it is a universal donor. There is no risk of coagulation or agglutination. Now this slide shows the different type of bloods and what are the genotypes of each type of blood and what are the antigens present on RBC of each blood type. A genotype of blood type A is IA, means both dominant genes, or IA. Genotype of blood type B is IB, or IBI. Genotype of blood type AB is IA, and that of blood type 0 and blood type O is small I. Now, IA is dominant, IB is also dominant, O is recessive. Now, this diagram shows antigens and antibodies in different type of blood groups. Group A blood has antigen A and antibody B. Group B blood has antigen B and antibody A. Group AB has both antigen A and B, no antibodies. And group O has both type of antibodies A and B and no antigen. Now this diagram shows the agglutination reaction. In this we can see that the serum of anti-A coagulates with AB blood type and also coagulates with anti-B. Type A coagulates with only serum of anti-A. Type B coagulates with anti-B, and in other cases there is no coagulation or agglutination, and when an antibody recognizes a antigen, it binds with it and a clumping is formed. Now let us see one more example of deviation of monohybrid ratio, not interaction, that is due to lethal allele in maize, which causes 1 is to 2 ratio instead of 3 is to 1. In corn or maize, the gene capital G is responsible for normal chlorophyll production. So seedlings with capital G and capital G small g are green, whereas with small g is yellow because no chlorophyll is formed, only carotenoids are formed. So seedling ratio is 3 to 1 if we consider green and yellow. That is the phenotypic ratio. The genotypic ratio is 1 is to 2 is to 1, that is capital G is to capital G is to small g. But after 10 to 15 days, endosperm exhausts, so yellow plants are unable to survive or die. So genotypic ratio becomes 1 is to 2. That is pure and hybrid because small g plants die. So the ratio of capital G and capital G small g, that is pure and hybrid, becomes 1 is to 2. In this case, monohybrid ratio is modified due to lethal alleles whose effect is sufficiently drastic, kills bearer of certain genotypes, that is small g, because it will not be able to form chlorophyll, so cannot survive further. Now, with this, we have explained incomplete dominance, and in the next class, we will discuss about the interaction of genes due to two different genes which affect the same trait.